Hello everyone, welcome in this channel and in this video I am Dr. Vijay Prasad speaking to you, and today's topic is a small intestine, anatomy and physiology of a small intestine. What is a small intestine? A small intestine is long muscular hollow tube which connects a stomach and large intestine. It is continuous with stomach to large intestine, and its length is about 25 feet. 25 feet, 6.5 to 7.5 meters. This, this is a small intestine. It develops from primitive gut tube. It develops from primitive gut tube, mid-gut portion of primitive gut tube. This is what is a small intestine. Now parts of a small intestine. Parts of a small intestine. Number one, duodenum. This part is duodenum. It is C-shaped structure. C-shaped structure. First part is duodenum. Duodenum, which is short. That is about 25 centimeter. 20 to 25 centimeter length is 25 centimeter. Then jejunum. Jejunum is about 250 centimeter, and then ileum. Ileum is about 350 centimeter. This is duodenum, jejunum and ileum. Three parts of a small intestine. First part is duodenum. It is C-shaped structure, and in this C-shaped pancreas is present in head of the pancreas is present in this C-shaped structure. This is pancreas. It is ducts open in the duodenum, and then it is continuous with jejunum. Continuity of jejunum is marked by duodenojejunal flexure. There is duodenojejunal flexure is there, but jejunum and ileum is not marked with any. They blend with each other. They fuse with each other. There is no demarcating line between jejunum and ileum. This is the parts of a small intestine. Now coming to the structure of a small intestine. The structure of a small intestine is like other parts of the GIT. Other parts of GIT in transverse section, we have seen that there is serosal from outside to inside, serosa, then muscular layer, outer longitudinal muscle layer, and inner circular muscle layer. Outer longitudinal and inner circular muscle layer, then inner circular muscle layer, then submucosa is there, and submucous layer, and finally there is mucosa. This is general structure of GIT. It is also in this case. There is slight differences between duodenum, jejunum and ileum. Now glands present in the small intestine are Brunner's gland. These glands are abundant in duodenum. It resembles pyloric gland, and Brunner's gland are there, and this number of Brunner's gland slightly decreases and finally disappear in jejunum. Brunner's gland, they secrete mucus and bicarbonate to neutralize the acidic kind. Then crypts of liver food. Crypts of liver food. These are simple glands and they secrete cells are present in this secrete enzymes and mucus. These two glands are specific in a small intestine. Then coming to cells. Cells present in these glands are number one, goblet cells. Goblet cells, Argentine, Argenti, Argentifin cells, Penneth cells, epithelial lining, epithelial cells are important cell in GI, small intestine. These goblet cells secrete mucus. Argentifin cells secrete secretin and histamine, 5-hydroxytryptamine, 5-hydroxytryptamine. Panneth cells and epithelial cells secrete enzymes. Some G cells are present in duodenum. They secrete gastrin. Then secretin cells are also present in duodenum and jejunum. These are the cells responsible for secretion of formation of intestinal juice. Secretion of intestinal juice, secretion of intestine is also known as intestinal juice or it is also known as succus entericus. Succus entericus. Succus entericus. Secretion of the small intestine is known as succus entericus or simply intestinal juice. Daily secretion is about 1.5 liters, 1.5 liter isotonic and alkaline in nature. Isotonic and alkaline is isotonic and alkaline in nature. 98.5% is water. Composition 98.5% is water and rest is 1.5% is organic and inorganic. Organic and inorganic substances. In this inorganic, potassium, sodium, bicarbonate, chloride, calcium, ions are present. In organic part, mucus, enzymes, and little amount of say hormones. These are present in succus entericus. This is composition of succus entericus or constituents of succus entericus. Now, in enzymes, first is introkinase. This introkinase acts on trypsinogen trypsinogen and convert it into its active form, trypsin. When active form of this trypsin auto-catalyzed the trypsinogen into trypsin, and it also acts on other pancreatic pro-enzymes into their active form. Trypsin acts on trypsinogen to convert it into trypsin, and in addition, it also acts on other proenzymes secreted from pancreas into its active form. Intracanal, then protein digesting, protein, ripsin, is a mixture of peptidases, aminopeptidases, dipeptidases, nucleotidases, etc. This is concerned with mixture of enzymes that is concerned with protein di- digestion. Then carbohydrate. For carbohydrate, maltase, many enzymes are present. Maltase, invertase, lactase, alpha dextrinase, and other enzymes are also present. For lipids, to digest lipids, lipase is there, then lecithinase, lecithinase, cholesterol esterase, these enzymes are present into the succus entericus. Now control of succus entericus. Succus entericus, no nervous control, neuronal control or nervous control and hormonal control. Hormonal control, that is humerus control or neuronal control, is not there. Its secretion is under control of local factors, then mechanical and chemical. Local factors, irritant causes increased amount of mucus. Local irritant causes increase in mucus secretion. Then mechanical is, that is distension of the small intestine. It increases volume and enzymes. Secretion increases volume and enzymes. Then chemical. Some food materials, chemicals present in, that is food materials, stimulate enzyme and volume secretion. Vagus stimulation leads to increased amount of volume and enzymes. VIP leads to increased watery secretion. So there is local factor, mechanical factor, and chemicals are present that controls secretion of a small intestine or succus entericus. This is control. Now, mechanism of secretion. Mechanism of secretion is not clear. Secretion is not clear. Sodium potassium pump, sodium potassium ATPase pump is involved in formation of this secretion, succus entericus. Mechanism of secretion is not very clear. Now coming to functions. Functions. Number one, mixing of chyme. Number two, digestion of food materials, protein, carbohydrates, lipid, cholesterol, phospholipids, nucleotides, nucleic acid, all are digested in this small intestine. Then after digestion, it causes absorption of digested material, end product of digested carbohydrate, lipids. This causes absorption of amino acids, glucose. Many types of glucose are vitamins are active process. But absorption of fructose is passive. Water absorption is also passive. Osmosis, due to osmosis, and then water and this lipids and its digested products are absorbed passively. This then absorption of digested material. Now protection of a small intestine by mucus and by alkali neutralized acidic chyme. It secretes immunoglobulin A, provides local immunity. Then helps in lipid, helps in control of lipid levels in plasma, helps to control lipid levels in plasma, maintain bile salt levels, and propel the chyme and undigested food material to large intestine. These are the functions of a small intestine, and bacteria present in GIT, including a small intestine, that is gut flora present in a small intestine, forms vitamin K and other B complexes and vitamin B complexes. Gut flora present in a small intestine causes formation of vitamin K and vitamin B complex. These are the functions of a small intestine and succus entericus. So for today, we learned about what is a small intestine, parts of a small intestine, the structure of a small intestine, secretion of a small intestine, and in secretion we learned control of succus entericus secretion and their functions and constituents of succus entericus. So thank you very much for watching. Please like and share this video and subscribe the channel. Thank you very much for watching. Let's meet again. Thank you.